Okay, let's start chapter 7, photosynthesis. Using light to make food. So nearly all life on Earth, including you, can trace its source of energy back to the sun. Nearly all, because there are organisms that live in the deepest parts of the ocean near the mid-crustal plates that are coming apart and that don't depend on the sun. It's warm down there, it's completely dark, and they have this really strange chemistry, metabolism, where they can feed off of inorganic stuff at the bottom of the ocean. They're really bizarre, but most of what the living things we think of, we're dependent upon the sun. Okay, electricity at photovoltaic solar panels produced from sunlight can be used directly, stored in batteries, transferred to the national electric grid. Living organisms that capture sunlight using photosynthesis can also serve as energy sources. Plant material can be burned. Plant materials can be used to produce biofuels in renewable energy. And so all these methods of capturing sunlight are renewable. And really, if you burn biofuels, if you burn plant material, what you're doing is using sunlight, captured sunlight, energy that was gotten from sunlight. And of course, sunlight is, well, it's inexhaustible. Right now, it is exhaustible. Eventually, the sun's going to run out of hydrogen. And then we're all, that's it. But it's not going to be for billions of years. So I'm not worried about it. Okay, so tapping into the sun, solar energy. Photosynthesis used by plants, algae, which are protistans, and certain bacteria. They're called cyanobacteria that are photosynthetic. We're going to learn that eukaryotes use a chloroplast to conduct photosynthesis. Bacteria don't have chloroplasts, but they can still do photosynthesis. I'm not sure how they do it without a chloroplast, but they do. Okay, transforms light energy into chemical energy. Remember, energy comes in different forms. So we're converting light energy into chemical energy, and chemical energy is really electrons, right? It's the electrons that form bonds between atoms. All right. Uses carbon dioxide and water as starting materials and releases oxygen gas as a byproduct. So is the purpose of photosynthesis to make oxygen? No, it isn't. It's to make sugar. Oxygen is actually a waste product, a byproduct or waste product of the process. And of course, that's how this oxygen got into the atmosphere, because of the waste product of photosynthesis. Okay, the chemical energy produced by a photosynthesis is stored in the bonds of sugar molecules. So that's the purpose of photosynthesis, is to capture the light energy into the chemical energy in sugar molecules. Okay, organisms that generate their own organic matter from inorganic materials are called autotrophs. Troph meaning nutrition, auto meaning self. So you make your own food. Plants and other organisms that do this by photosynthesis, called photoautotrophs, are the producers for most ecosystems. Now, they're, like I said, those bizarre organisms that live at the bottom of the ocean are called chemoautotrophs. They store energy in a completely different way and not using photos. So they're chemoautotrophs. But plants and other organisms like the cyanobacteria and algae are called photoautotrophs. So the fact that nearly all organisms can trace their source of energy back to the sun clearly illustrates that this ability to transform energy in matter is vital to the existence of life on Earth. Okay. And there's the photosynthetic autotrophs. Plants, mostly on land. Photosynthetic protists, algae, which are mostly, in water, either freshwater algae or marine algae. And then photosynthetic bacteria, cyanobacteria. So actually, when we do the algae ID lab, you'll learn that algae is not a real specific term. It includes organisms from completely different kingdoms. It includes protestin algae that are in protestin are eukaryotes. And it contains bacterial algae, the cyanobacteria. And that's one of the things you'll use in the algae ID lab to distinguish a protestin algae from a bacterial algae is the protestin algae are going to have chloroplasts, and you can see them, and they're often different shaped. That's one diagnostic characteristic of them. While as the photosynthetic bacteria are just like uniformly green, and you can't see any internal structure because they don't have chloroplasts. So most ecosystems depend on a process of photosynthesis to convert the energy of sunlight into high energy organic molecules, which then provide fuel for organisms, which major theme, this is pathways that transform energy to matter. Good. So chloroplasts are light-absorbing organelles. The site of photosynthesis and found mostly on the interior cells of leaves. Their green color is from chlorophyll, a pigment. It's a light-absorbing molecule in the chloroplast that plays a central role in converting solar energy to chemical energy. It's really like a light antenna. It captures light and converts that light energy into the energy of electrons. And then those electrons are moved around in the chloroplast in a internal membrane of the chloroplast. Stomata are tiny pores in leaves where carbon dioxide enters and oxygen leaves. So they're tiny and usually underneath the leaf. So they get the carbon dioxide from the atmosphere, and the oxygen that's made as a waste product goes out through these same little tiny holes. Membranes within the chloroplast form the framework where many of the reactions of photosynthesis occurs. So a lot of these, and we're going to see chemiosmosis again. We're going to see proton pumping to make ATP. Very similar, actually, to what's happening in mitochondria, because making sugar molecules from carbon dioxide. So the sugar, glucose is C6H12O6. Those 6 carbons and those oxygens came from carbon dioxide. So what's called fixing carbon dioxide. That takes a lot of ATP to do that because it's work. You're making a complicated molecule, glucose, from a very simple molecule, carbon dioxide. So that's work, and it requires ATP, actually a lot of it. So first, in photosynthesis, you got to make ATP, right? And higher energy electrons. And then you can use that ATP and the higher energy electrons to convert carbon dioxide into sugar. So we're going to break the photosynthetic process, like we broke cellular respiration, into pieces. We're going to break photosynthesis, which is a complicated procedure, into 2 parts. Okay, journey into a leaf. Okay, there's the stomata on the underside of the leaf. These are photosynthetic cells. This is sometimes called the mesophyll, the inside of the leaf. There's the cell, the mesophyll cell. There's the chloroplast, and then below the chloroplast, you have the inner and outer membranes, like a mitochondria. And you have these stacks called grana, granum plural, granum singular, and the thylakoid membranes in a thylakoid space. And the capturing of light, the making of ATP happens in this thylakoid membrane. So like a mitochondrion, as I mentioned, chloroplast is a double membrane envelope, The inner membrane closes a compartment filled with stroma, a thick fluid, so it's stroma, and part of photosynthesis happens in the stroma. Suspended in the stroma are interconnected membrane sacs called thylakoids. The thylakoids are concentrated in tracts called grana, and the chlorophyll molecules that capture light energy are built into the thylakoid membranes in what are called photosystems. In the overall equation for photosynthesis, notice that the reactants of photosynthesis, carbon dioxide and water, are the same as the waste products of cellular respiration. And that photosynthesis produces what respiration uses, glucose and oxygen. So this is the recycling. I think we saw this in the energy chapter, how energy is recycled in the biosphere, that plants take CO2 and water with sunlight to make glucose and oxygen. And then other organisms like heterotrophs take glucose and oxygen to make ATP and give off carbon dioxide and water, which the plants can use. And we got a cycle. But remember, the plants are also using their own glucose, eating their own glucose, and using their own oxygen at night and in the winter. So remember, plants have both mitochondria and chloroplasts. Unfortunately, they make more oxygen than they use. So there's a net gain of oxygen to the environment. Because us heterotrophs, we only have mitochondria. So we have to use the oxygen in the atmosphere to break down the glucose that we get from the plants to make ATP to keep ourselves alive. Okay. There's the overall view of photosynthesis. 6 carbon dioxide plus 6 water, light energy to make glucose and oxygen gas as a waste product. That's the overall equation. So here's the way we can divide photosynthesis into 2 stages connected by energy-carrying and electron-carrying molecules. First is going to be the light reactions, in which chlorophyll and the thylakoid membranes absorb solar energy, which is then converted to the chemical energy of ATP and a molecule called NADPH. And that should look very familiar to you. Well, ATP is familiar to you, but NADPH looks a lot like NADH, right? And what is NADH? High energy carrier molecule used in cellular respiration. Photosynthesis also has high energy carrier molecules, but it's NADPH. Whereas NADP is reduced to NADPH and carries high energy electrons over to the Calvin cycle, number 2, along with ATP. So the light reactions make ATP and NADPH. They're sent over to the Calvin cycle, which uses the products of the light reactions to power the production of sugar from carbon dioxide. So again, we're going to need to know the input and output molecules of these processes, just like you had to know the input and output molecules for the parts of cellular respiration. What's the input molecule for glycolysis? What's an output molecule for glycolysis? So on and so forth. And here's the roadmap. So we got water coming into the light reactions in the thylakoid membrane. Now, Calvin cycle, I believe, happens in the stroma. The light reactions happens in the thylakoid membranes. So water comes in and light. Those are input to the light reactions. And an output from the light reactions is oxygen. And where did this oxygen come from? The water that came in here. This oxygen right here are these oxygens down here. So what plants do is they break water open. And the water that comes into the plant, the oxygen in the water that came into the plant, leaves the plant as O2, as molecular oxygen. Right? And we'll see that water supplies replacement electrons for photosynthesis. So break water. So in cellular respiration, you make water. In photosynthesis, you break water. And see here, the light reactions make ATP, make a lot of ATP and NADPH, carriers of high energy electrons, and they go over to the Calvin cycle. Input molecule for the Calvin cycle here is CO2. And it comes in here and goes around, and 6 of these are put together using the ATP from the light reactions and the high energy electrons from the light reactions to convert CO2, 6 CO2s into 1 sugar molecule. And then ADP and PI are sent back over from the Calvin cycle to the light reactions and ADP, and it goes around and around like this. And an output molecule for the Calvin cycle is sugar, is glucose. So initial incorporation of carbon from the atmosphere into organic compounds is called carbon fixation. So you're fixing carbon. And this helps reduce the concentration of carbon dioxide in the atmosphere. So it helps with global warming. So it's good to have all these huge forests all over the globe, because the more photosynthesis is going on, the more CO2 is being taken out of the environment, and helps to reduce the amount of CO2, which is a greenhouse gas. So relationship between photosynthesis, carbon fixation, and global climate, good example of how interactions between biological components and many different levels affect all life on Earth, because climate change is going to affect all life on Earth. So what molecules are the inputs of photosynthesis? What molecules are the outputs? Input. Water is an input. CO2 is an input. What molecules are the outputs? Glucose and oxygen are the outputs. But remember, I could also say, what are the inputs to the light reactions and the outputs of the light reactions? What are the inputs to the Calvin cycle and the output of the Calvin cycle? So water is really input for the light reactions. And ATP and NADPH are output molecules along with oxygen. Input molecules to Calvin, CO2, and ATP and NADPH from the light reactions. And the output molecules are sugar, glucose, and ADP and PI plus an ADP that goes back to the light reactions. So deforestation in Asia can alter the climate in North America, because like I said, all these forests taking CO2 out of the atmosphere, and now you cut them all down, and you reduce you raise the amount of CO2 in the atmosphere by doing this. So this is interactions within biological systems. The light reactions, converting solar energy to chemical energy to the nature of sunlight. Sunlight, like other forms of radiation, travels through space as rhythmic waves. The distance between the waves of crests of 2 adjacent waves is called a wavelength. And this, the symbol for wavelength is an upside down Y. It's a Greek letter. So, and so we talk about electromagnetic radiation as what wavelength it has. And the wavelengths can be incredibly tiny, like billionths of a meter, or it can be actually meters or kilometers wide. And this is the full range of radiation. Is called the electromagnetic spectrum. And there it is. All the way from 10 to the -5th nanometer, which is like, must be like a trillionth of a meter, all the way up to 10 to the 3rd meters, 3,000, kilometers. And there's a wavelength right there, from crest to crest. So light is really an odd beast. It comes at us both as particles and as waves, because these particles are called photons. And remember, they're little bundles of pure energy. But the photons come at us in waves of different wavelengths. And the relationship is, the smaller the wavelength, the higher the energy, of the photon. So photons are really tiny wavelengths, are actually really dangerous to living things. We're talking about gamma rays and X-rays, and to a certain extent, ultraviolet radiation. It's dangerous. It will kill you in short order. That's why it's not a good idea to have lots and lots of X-rays. All right? And that's why the radiologist, when you go in and have an X-ray done, he or she sets you up to wherever your X-ray, what part of the body your X-ray, he or she walks behind a lead shield to push the button to turn on the X-ray. Otherwise, that person would be exposed to hundreds of X-rays over the course of the day, and they're damaging. They destroy cells, and gamma rays are even worse, even more dangerous. And here's the thing: the sun sends us the whole electromagnetic spectrum, the whole thing, including gamma rays, X-rays, UV, and what's called visible light. Fortunately, our atmosphere filters out the gamma rays, all the X-rays, and most of the UV. A little bit of the UV gets through. Right? That can cause sunburn and possibly skin cancer. And then getting to wavelengths of about 350 or 380 to about 750 is what's called visible light. It's called visible light because it's what we use to see with. That's the light that our eyes are sensitive to, that we can see. But it's also the light that can be used in photosynthesis. Right? And let's say from 350 nanometers to 750 nanometers. Also, that's the whole range of colors, like RGB, all the way from violet to blue to green to yellow to orange to red are in there. And so all these colors are different wavelengths. All right? And we're going to see that and we're going to do this in the chromatography lab. When a photon hits a molecule, only two things can happen. It can bounce off the molecule. This is called reflected light. Or it's absorbed by the molecule and becomes, and fires up some electrons within that molecule. And sometimes that energy is giving off as heat after the molecule, after the photon is absorbed by the molecule. So here's the thing. What photons are used by plants in photosynthesis to make sugar? Do we use all of the spectrum, all the way from 350 to 750? Or do they just use some of the wavelengths? Well, I'll tell you, they only use some of them, because just by looking at plants, you can tell which ones they don't use. And that's right around 550 nanometers, because that's green. Because plants do not use the green photons. How do I know this? Because they're green. The color you see is reflected light. All right? So they are completely reflecting the green photons. So they're not using them. Which ones are they using? Well, you can't just look at the leaf and tell which ones they're using. So in lab, we are going to shine coherent light into chlorophyll and another photopigment and find out what photons they are absorbing. Because the photons used in photosynthesis have to be absorbed by molecules, by the chlorophyll molecules, not reflected. So we're going to find out in lab which ones are absorbed and therefore which ones are used. And then later on, if you go ahead to make spectrum slightly higher larger wavelengths are called infrared, and those are really just like heat waves. And then microwaves for cell phone communication, and then radio waves, which is the lowest energy, and they have wavelengths of tens of meters and kilometers even. Okay. Okay, so in 1883, German biologist Theodor Egmont saw certain bacteria living in water cluster in areas with higher oxygen concentrations. So here's a guy who figured out what wavelengths of lights were being used by photosynthesis by using bacteria that cluster around where oxygen is being made. Because photosynthesis gives off oxygen, right? So the bacteria are going to cluster around where the oxygen is being made. So he placed freshwater algal cells within a drop of water on a microscope slide. And most bacteria congregated on algae exposed to red, orange, and blue-violet light. So there it is. And we're going to show this in a much more sophisticated way using an instrument called the spectrophotometer. In 1883, they didn't have this instrument. But this guy showed which wavelengths of light are being used in photosynthesis by spreading out the light. Remember, white light, is the whole spectrum, all the colors of the spectrum. And you can divide that by using a prism. So we used a prism to divide the white light into all the colors. And showed all those colors on a slide, and then put oxygen thriving bacteria on the slide. And where did he see the bacteria congregated? Right around 425 and right around 600 or 650. So those are actually the areas, the wavelengths that are mostly absorbed, maximally absorbed by chlorophyll. And therefore, they're the ones that are used to make sugar in photosynthesis. Notice that there are almost no algal cells in the middle where the green photons are, because, like I said, green photons are not used in photosynthesis, so there wouldn't be any oxygen there. Okay. So selective absorption of light by leaves explains why they appear green to this. Light of that color is poorly absorbed and is reflected, and therefore that's why you see that color. So remember, white is the reflection of all the colors. So like white clothing would be, means that all of the photons are being reflected. That's why you see white. Black clothing, all of them are being absorbed. And that's why they appear black. So, and in clothing, the photons that are absorbed are reflected back out as heat. So you don't see them as colored, but they come as heat. So on a nice hot summer day, is it better to wear white or black clothing? Well, better to wear white. It's reflecting all those photons, and so your clothes aren't heating up. On a nice cold winter day, better to wear black clothing. All the photons are being absorbed and they're being re-radiated as heat. So your clothes are actually helping to heat your body if you wear black clothing in the winter. Energy cannot be destroyed, so the absorbed energy must be converted to other forms. And in clothing, as I mentioned, it's converted to heat. And actually, and eventually, remember, all of the energy that comes into organisms eventually leaves as heat. So you can't create energy, you can't destroy it, you can just transform it. And you can warm yourself by it, and you can use it to do work, but eventually leaves as heat. So that's why leaves are green, because they are reflecting the green photons. They're not absorbing them. Okay, chloroplasts contain several different pigments that absorb light at different wavelengths. And we're going to separate these in a lab, in a chromatography lab. Chromatography is separating mixtures of molecules. So there's two types of chlorophyll, chlorophyll A and chlorophyll B. And we are going to separate them in lab. And then there's also a family of yellow-orange pigments called carotenoids, which absorb mainly blue-green light, but they're and they reflect yellow, so they are yellow pigment. And we're going to separate those in lab also from spinach leaves, chlorophyll A, chlorophyll B, and a carotenoid. There's also called anthocyanins, which are the red pigments. But spinach leaves don't have any of those. But maple leaves do, and that's why we see the fall colors. For some reason, these trees, mostly maple trees, they take in the chlorophyll back into the trunk in the fall and save it. And they throw away the carotenoids and the anthocyanins. And that's the yellow and the reds. That's why we see the fall colors, the yellows and the reds in the fall, because the chlorophyll has been, and during the summertime, in the spring, there's a lot more chlorophyll than there are carotenoids and anthocyanins. So that's why they're green. But when the plant takes the green chlorophyll in, that's when you can see the yellows and the reds. So there's the fall colors, because the yellow and the red pigments are being thrown away and the chlorophyll is being saved. So, well, this is reiterating what I just mentioned. Light behaves as waves and particles at the same time, called photons. The shorter the wavelength of light, the greater the energy of a photon. When a pigment molecule absorbs a photon, one of the pigment's electrons gains energy and is now said to be excited. So the energy of photon is absorbed as the energy of electrons. And remember, those two things are sort of similar. They're both little discrete packets of energy, photons and electrons. Photons can exist by themselves out there hitting you. Electrons never do that. Electrons are always associated with an atom and in molecules. Okay. An excited electron usually loses its excess energy and falls back to its ground state. And what it does is it releases heat. So this is what happens when the pigment molecules in your clothing absorb certain electrons and absorb certain photons and excites electrons in those pigment molecules. And then those electrons fall back to the ground state and release the energy of the light out as heat. That's what I was saying. You wear black clothing, and you're really absorbed. You are going to be you're going to be warmer, because all of the pigment molecules in the black clothing is absorbing all the photons and releasing all of them as heat. Whereas white clothing, none of them are absorbed. They're all reflected. So it'd be a lot cooler. Okay. Some pigments emit light as well as heat after absorbing photons, and this is fluorescence. So you've seen the glow sticks, where you break them and shake them, and this is called a chemical reaction that excites electrons of a fluorescent dye. The excited electrons quickly fall down to the ground state, releasing energy in the form of fluorescent light rather than heat. This is also what happens in fireflies. This is called actually cold light. Usually, light bulbs get really, really hot, but the rear ends of lightning bug doesn't. This is called cold light. And of course, the glow sticks, they don't get hot. That's just called cold light or fluorescent light. So there are the glow sticks. So we have a chlorophyll molecule, light hits it, excites it up, raises the electron to an excited state. An electron falls back to its ground state. If it's just chlorophyll in a solution, like in the lab, it will release its energy as heat, and fluorescence lights. But in the leaf, it's going to send those excited electrons onto another molecule, an electron acceptor molecule. In the thylakoid membrane, chlorophyll molecules are organized with other molecules into photosystems. Each photosystem has a cluster of a few hundred pigment molecules, including chlorophyll A, B, and some carotenoids. And this cluster of pigment molecules functions as a light gathering antenna. Right? So there is a cluster of pigment molecules, a photosystem within the thylakoid membrane. So the thylakoid membrane is phospholipids, and you have a group of proteins in there. And a cluster of pigment molecules. The chlorophyll A and B and the carotenoids. And notice that the photon is absorbed by one of the chlorophyll and it's passed along to electron acceptors. This fired up electron is passed around to electron acceptors. And after this electron leaves, it leaves an electron hole in the pigment molecule. So that's where water comes in. You got to replace that electron that got excited and left. So what you do is you break water apart and take electrons from the broken water molecule and replace that. So another photon can come along and raise that one to excited state, and that one can be passed along. So the reactions of photosynthesis are driven by membrane-bound enzymes, and the folded shape of the thylakoid grid increases the area on which these enzymes can operate. So which major theme is this? Relationship of structure to function. We have these folded membranes with a lot of surface area to absorb a lot of light and make a lot of fired up electrons. So two photosystems cooperate in the light reactions. Photons excite electrons in the chlorophyll of the first photosystem, energize electrons from the first photosystem passed down electron transport chain to the second photosystem. So we're going to have electron transport again. And this is going to be chemiosmosis all over again to make ATP. So really, chloroplasts make ATP in almost exactly the same way that mitochondria make ATP by electron transport. The chloroplast uses the energy released by this electron fold to make ATP. The second photosystem transfers its light-excited electrons to NADP, reducing it to NADPH. So the first photosystem makes energized electrons that can be passed along to pump protons across the membrane to make ATP by a chemiosmosis. And then those electrons become tired again. A second photosystem absorbs a second photon to fire up that electron a second time. So it can be passed on to NADP to make NADPH. And there it is. So the reaction center chlorophyll here absorbs a photon, fires up an electron. That electron is replaced by breaking of water and putting electrons back in there. So this primary electron acceptor falls down this electron transport chain, and it doesn't show it here, but protons are pumped across the membrane, just like in mitochondria. And a lot of ATP is made. Then this electron becomes tired, can't be used to pump any more protons. It enters a second photosystem where another photon is absorbed to fire it up here so that these fired up electrons can be passed on to NADP, reduce NADP to NADPH. And this ATP and this NADPH, they're the output molecules from the light reactions, are going to be sent over to the Calvin cycle, right? Those are going to be input molecules to the Calvin cycle. So the light reactions are located in the thylakoid membranes, the two photosystems, and electron transport chain that connects them, transfer electrons from water to NADP, producing NADPH. And there is the how the thylakoid membranes converts light energy to the chemical energy of NADPH and ATP. Here is electron transport, pumping electrons. And you have ATP synthase, just like in mitochondria. So you're pumping hydrogen ions from the stroma into the thylakoid space. Those hydrogen ions flow back through ATP synthase to make ATP, exactly the same as mitochondria, right? And notice that these NADPH and ATP is sent over to the Calvin cycle. Why is water required as a reactant in photosynthesis? As replacement electrons for the electrons fired up in chlorophyll. They have to be replaced. So they can be fired up and passed along also. So water is a source of replacement electrons for the photosystems. So first photon fired up, falls down, electron transport, another photon fires it up to send it over to NADPH. The same thing as the earlier slide. So the mechanism of ATP production during the light reactions, as I keep on saying, is very similar to the mechanism we saw in cellular respiration. In both cases, an electron transport chain pumps hydrogen ions across the membrane. ATP synthases use the energy stored by the hydrogen gradient to make ATP. Exactly the same. The main difference is that food provides the high energy electrons in cellular respiration, where light excited electrons flow down a transport chain through photosynthesis. So the fired up electrons are in food in cellular respiration, are taken out from food. The fired up electrons in photosynthesis are fired up by light to make the food. Okay, so then we're going to go into the Calvin cycle. So I'm going to break it right here. And in the next video lecture, we'll talk about the Calvin cycle. So this is going to be 106 photo 1. So I'll see you later on that one.